All right. Hello, everyone. Thank you for coming to this edition of the MPG Primer. My name is Geraldine van der Auwera. I'm representing the data sciences platform today. I want to thank you all for coming this early. I know it's pretty early go time. I expect that there will be a few more people filtering in, but to respect everybody who made it on time, I want to get started without any further delay. Okay, so today I'm going to be talking about the high throughput sequencing and variant discovery pipelines that we have here at the Broad, which are also known as the GATK best practices workflows. So my goal is to give you an introduction to scientific and technical background that you need, as well as give you an overview of the pipelines, the analysis pipelines themselves. Better if I remember my clicker. All right, so I want to say, as I start, I'm representing the data sciences platform. We are twinned with the Genomics platform, which is includes the sequencing facility into an entity within Broad that's called Broad Genomics. This is only a small sampling of the people who are involved in this effort. And I want to reiterate that what I'm going to be talking about today is the result of the work of dozens of people working really hard. So I want to acknowledge their hard work. Okay. First, a quick prelude. I'm going to talk a little bit about the goal of what we're trying to achieve with these pipelines. Okay. We're trying to ultimately discover variants relative to a reference genome. And by variants, I mean genetic changes in individuals that are relative to a reference genome. The reference genome is an artificial sequence, haploid sequence, that is based on the consensus from data from many individuals, and that attempts to represent the average sequence of a human. Now, obviously, given the diversity of human populations, this is a fairly tall order, but it's as standardized as possible to provide a common frame of reference. Over the years, there has been some development, some addition of complexity to the human genome reference that has aimed to better represent some of the sequences that we find in human populations that are traditionally not well represented in this field. And so in addition to the main sequence, you will have within the reference areas that are called alternate contigs, which attempt to represent haplotypes from these populations, so that when we do a variant analysis on individuals from these populations, we can more accurately represent their particular variation. Now, this is something that, even though it's been available for a few years, not all our software takes full advantage of the presence of these alternate contigs. And our default production pipeline actually does not take full advantage of that, but there's a development in the works to improve that situation. But it is also always possible for interested researchers to take advantage of that if they are studying populations for which this applies. All right. So I'm going to focus, by the way, very much on humans, because that tends to be our focus here at the Broad. If you are working on other organisms, you do need to have a fully assembled reference to work with to do variant discovery. These are available for many organisms. Some they're not. In the bacterial world, microbial generally, things are a little bit complicated. You can have draft genomes and so on. It's still possible to run these analyses, but it takes some additional technical workarounds, complexity. I'm not going to go into the detail on that, but there's documentation available on the GATK website for people who are in that case. But since we're talking about human medical and population genetics here, I will focus on the humans. I should also add that when we're looking for genetic differences in individuals, we will break down different categories of variation. The first big split is between germline variation and somatic variation, where germline is the inherited variation, or the variation that's present in the germline cells generally. Whereas somatic variations are typically, we often mean cancer by that, but there are somatic mutations that are not cancers, of course. During the course of our life, we accumulate many somatic mutations that never go on to cause what we call cancer. But later, when I talk about the somatic pipelines, I will focus mostly on cancer because that is the topic of study that is most represented here. All right, so the other big thing you need to be aware of in terms of background setup for these analyses is that the experimental design is going to have a really big impact on your experience working with the data, and also the kind of information that you can derive from the data. And here I want to contrast mainly whole genome sequencing from exome sequencing. And I'll fold gene panels into exome sequencing as they're technically similar in terms of the difficulties. Okay, so there's a lot of debate about whether you should always have whole genome or whether exome is sufficient. What it comes down to is that if you're sequencing the whole genome, by definition, you're hoping to capture sequence across the entire genome of your subject. Now, we know that technically there are areas of the genome that we have a hard time sequencing, for example, the telomeres, the centromeres, and other subsets of chromosomal regions that are just difficult to get to with sequencing techniques. But for all intents and purposes, you do get sequence coverage across all of the usable sequenceable genome. And what that empowers you to do, of course, is you can identify genomic variation in any region, whether it's within genes, within exons, within introns as well, but also outside of the gene context. And so the whole genome gives you a huge amount of data and does not limit you in terms of where you can identify variation. So in principle, that's great. The downside of that is that it represents a huge pile of data, and that can be fairly overwhelming and cause scaling difficulties. I'll talk a little bit about that a little later on in my talk. In contrast, also it's more expensive. In contrast with the exome sequencing or gene panels, there you're restricting the territory that you're sequencing to a much smaller subset. So the amount of data you're working with is more manageable, and it's definitely more affordable. But there are two problems with exome sequencing. One is that you limit your ability to identify variation in regions that are not explicitly targeted by whatever kit you're using to extract the sequence. So that's a big limitation. And the other thing is that from a technical standpoint, the processes that are used for extracting exome data are prone to various types of technical biases, which whole genome sequencing is not. And so from a data analysis perspective, we prefer whole genome just because there are fewer weird technical biases that we need to deal with. But depending on your goals, depending on whether you know what you're looking for or not, you need to weigh these different factors and consider what is best for your particular study. All right. By the way, feel free to interrupt me at any point if you have questions. Okay. Now, the other distinction and piece of context to really understand what's going on with variant discovery is that we classify genetic variation based on the size and type of difference, right? And here we distinguish short variants. Short variants are going to be your SNPs and indels. SNPs stands for single nucleotide polymorphism. That's a one single base substitution. We call it a polymorphism in the germline context. We'll call it a variant in the somatic context, but it refers to essentially the same thing at the in terms of what the genetic change is. And you have indels, which are insertions and deletions, and those are small stretches of DNA that are either missing or added in the individual that you're studying relative to the reference. These are short variants that GATK, which is a tool I'll introduce in a few minutes, has traditionally specialized in, but there are also, and these are fairly tractable, these are well understood in terms of how we do the variant discovery. There are other types of variants that are a little more complicated. Copy number variation and structural variation, especially, tend to be bigger changes that, in the case of copy number variation, you can have either additional copies of the genetic region, or you can have a structural variation where you have inversions, translocations. Those genetic stretches have been moved around. These are a little less well characterized, and there's still quite a lot of development work that's going on for us to really do a good job of identifying them. We have some really solid tools, which I'll be talking about, but it's important to understand that there's still a fair amount of work that needs to be done to achieve your perfect sensitivity in terms of being able to identify these variants, just because by nature they're a bit more complex. All right. This is a type of visualization of variation and sequence data that will come up several times in the presentation. So I just want to introduce what it looks like. This is an IGV screenshot, another tool I'll introduce in a minute. And it's showing us reads. The gray bars are individual sequence reads. And that's showing us that at a position where we have a difference in our individual relative to the reference, in some of the reads, this is shown up such. Here we have a probable, very likely C to T SNP that's showing up very clearly. This is a very clear site. We have nice coverage. There's good quality. And we have a good balance in terms of representation of alleles. But you have to understand that most of the time, it's actually the sequence data is quite a bit messier than that, because there's various reasons why errors creep into the sequencing process. And so, while this is a very clean case, a lot of the cases we're going to be dealing with and struggling with are the cases where the sequence data is messier, and we have to use some sometimes fairly complex algorithms to be able to differentiate the noise from the actual signal. All right. Now, for short variants, we have a standard format for outputting them, which is the VCF, variant call format. This is just a text file where you have a header that summarizes some amount of metadata about your sample, your cohorts, and the annotations that have been added to the data. And then you have a description that is, and I'm not going to go into detail, but just to introduce to you to this format where we're inventorying each variant position, each variant based on its position in the genome. So the chromosome, the position on the chromosome, whether there's been any previous reports of this variant, and then all the information about the alleles in this area. Then finally, you have, for each sample, if you're looking at multiple samples, for each sample, you have the genotype and statistics associated with the genotypes. So that's the generic, the standard format for working with short variants. For the other types of variants that I've mentioned, copy number and structural variants, there is not a standardized format yet. They can, to some extent, be represented in VCF, but there is much less consensus among tool developers. And so the representations are a little less standardized. Okay. So that was just the background information of what do I mean when I talk about variant discovery, and a few technical things you want to have in your pocket just to understand what's going on. Okay. Now, for the actual meat of the primer, what I want to go over are these 4 sections. First, I'm going to very briefly introduce the tools that we're going to be using so that when I throw out these names, you know what I mean. I want to give you a quick introduction about the sequence data itself, how it's generated, and what are the formats that you're going to be working with. Third, I'm going to talk about the pipelines themselves, what goes on in the workflows, what's processing happens is done to the data to actually derive the variant information. And finally, I want to do a quick note to introduce an analysis platform that was built in data sciences platform, which you may have heard of, which is called Firecloud. And I'll give you just a few pointers about that platform, as I believe many of you may have the opportunity to use it in your own work. All right, any questions at this point? No? Okay. All right, so let's briefly go over the tools. I'm going to talk about the Integrative Genomics Viewer, IGV, which is the genome browser that was originally developed at the Broad by Jim Robinson and team. There are many other and several other major genome browsers. You're welcome to use whatever you feel comfortable with. We work with IGV because there's a lot of functionality that's specifically developed for visualizing things we care about in terms of the variant discovery pipeline. For genome mappers, I'm going to talk about 2 mappers. BWA, which was written by Hung Lee here at the Broad, which is a majority aligner for DNA sequence. And also the STAR aligner, which is the aligner we use for RNA-seq. RNA-seq has to be handled a little bit differently in terms of the mapping because of the presence of splice junctions and so on. The Picard tool, or toolkit, I should say, is a toolkit also developed here at the Broad, but it's also an open source community project, so there are contributors outside of the Broad as well. We support these tools on the GATK forum, so if you use them, you have problems, please come to the forum and we'll help you. And that toolkit has mainly to do with processing of the initial sequence data, as well as quality control. There's a lot of very useful quality control tools in Picard. And finally, GATK, Genome Analysis Toolkit, which is kind of the leading variant discovery software package, also developed here at the Broad, developed within a data sciences platform. And it was previously licensed for commercial use, but now, starting with version 4, which is coming out soon, that is completely free and open source for everyone. Okay. A quick note about the new version, in case you've already worked with GATK in the past. GATK 4 has been completely rewritten from some aspects to improve scalability, to improve speed and performance. We've also added a number of analysis tools to be able to identify some variant types that we previously weren't able to do. And we're integrating the Picard tools to make it easier to work with both toolkits. And we are planning to release this new version on January 9th, 2018. Now, when I say more tools for more variant types, previously we specialized in germline SNPs and indels, but we've expanded to covering copy number variation on both the germline and somatic front, as well as short variants in the somatic front as well. MUTEC2 is based on the MUTEC tool developed originally in the CGA group by Gadi Getz's team. And we're quite excited to have this toolkit out. It's available as a beta version right now. The key takeaway is that we now have best practices workflows for a lot more use cases. All right, so let's talk about the data, how we generate it, and how we work with it. I won't go into the particulars because there's a lot of different ways to groom this particular cat. But the basic idea is that you have some kind of biological sample that comes from a subject. You're going to extract DNA from that sample or RNA. If you're extracting RNA, you're going to have some kind of processing step to make complementary DNA that we can actually sequence. We can't sequence the RNA directly. Once you have your usable purified DNA, you're going to create fragments that are going to then be processed by several reactions in vitro that mainly add things like barcodes and adapters. These are all technical appendages that are used for the sequencing process, right? Now, the really important thing here is that we come to the concept of read group. Whatever unit you have of DNA that you're making, this is producing a library prep, that produces a unit of sequence as far as we're concerned for the analysis. So the library of fragments of DNA is a foundational concept for us. Okay. The second part is you have that library. Yeah, go ahead.
During library prep, how exome and genome differ, and how it is exactly that they isolate out the exome sequences without even knowing sometimes where they all are.
So what's the big difference between whole genome and exomes? Again, for exomes, there's various different ways of producing an exome library. But I would say the major difference is that with whole genome, you're taking everything, you're chopping it up in fragments, and you're pushing that through. With exome, what you're going to do is you're doing an isolation of the fragments that overlap with exon sequences. And that's typically done using something like baits, which are essentially molecular probes. They're kind of like PCR primers, but you're using them to capture those fragments specifically and filter out everything else. So part of the process involves designing baits to capture specifically what you want. And that actually has an important consequence because depending on the manufacturer, those sequences will be different. What you're capturing is actually slightly different. So across manufacturers, across their kits, you will capture different territory. Most of it will be in common, but there will be differences. And that has consequences for when you're comparing exome sequencing results that were done in different places. If they were done with different kits, you can only really compare the intersection. And you also get into a weird place sometimes with technical biases that affect the amount of coverage you have in certain regions. So that's one of the reasons we don't like exomes, because whole genome is a lot more standardized. Like you just get everything. It's easier to deal with. Okay, that was a good question. Thank you. All right, so we have our library. Now we're going to sequence it. We mostly use Illumina technology here. We're going to have something that looks like this. Depending on the model, it might look different, depending on the model of the machine and so on. But you have a flow cell, which is the chip. You're going to load your samples, your libraries, onto lanes of the flow cells. They're physically separated channels. And from one lane, that's where the sequencing process will happen. Now, Illumina has some excellent videos that explain the chemistry and the actual sequencing process. The idea is basically you shooting lasers at the molecules and identifying based on the color that comes back which bases we have in these little clusters. Watch their videos if you want to learn more about the sequencing process. What's important here is to understand that there are two things. One is that the sequencer will sometimes make mistakes because there's a lot of things that can influence the result and make the reading a little fuzzy. So the readings that we get off of the machine are not perfect, and there's a little bit of room for error there. And the other thing is that if you look at the libraries that were sequenced on a lane, typically we multiplex libraries on lanes, meaning that we will put DNA from multiple libraries, which have been barcoded, so we can identify reads separately. But we'll put several libraries on the same lane, and we'll put aliquots of the library across multiple lanes. So you end up with a crisscross matrix of your reads that are spread across this lanes of this flow cell. So one of the things that we're going to have to tease apart in the first stage of the analysis is recognize which read belonged to which library and so on. And that's going to be part of the data pre-processing. Okay. Now, once we have the raw sequence, it's typically delivered in FASTQ format. Most of the world uses FASTQ. At the Broad, we actually don't look at the FASTQs. We don't care about FASTQs. We don't like that as a format. And so we have a slightly different workflow. But I do want to tell you what is the standard. And the formatting of the information is the same. The key thing is that when you get your read sequence, you have this information. You have the sequence name, which is composed by the machine based on some rules. You have the sequence itself, the sequence which was read off of the fragments of DNA. And you have base quality scores. And this is a super important point, that for each base that was read, the machine emits a quality score, which is its estimate that it may or may not have made a mistake there. It's its level of confidence in that base call. And so it's encoded in this weird format, which is ASCII, which is just an encoding that allows us to represent a value like 34 as a single character so that we can have a one-to-one correspondence. The values themselves are on something called the Phred scale. And the Phred scale is basically a log transformation that allows us to represent these confidences, these probabilities, on a scale that is more humanly intuitive. Because if you think about how you have a difference between 90% confidence and 99% confidence, the 99.9, it can be difficult for our brains to really gauge how much of a difference it is when it's just 0.9 difference. But when we put this on this log scale, when you go from a Q20 to a Q30, it gives you more tractable resolution there to be able to differentiate how much of an impact the difference has. And it's also much easier to represent in the files, so that instead of having a lot of decimal places for each number that we need to store, we can just store it as an integer. Okay?
Practically,
yeah?
what does 3, 7, 8, 8, and semicolon represent in?
So these are each going to represent a Q value. So one of these might, and forgive me, I forget the exact encoding, but this might represent 20, this might represent 30, things like that. So that's the idea of the score. Or 20 or 25. Okay?
Yeah, I see.
Yeah. And like I said, it's really a way to have a multi-character something in a single character, right? Okay. So that's how we generate the data, and at that point, we have a big pile of reads that is encoded in a somewhat cryptic format, and we somehow need to extract variance calls from that. So next step, the pipelines. We call them the best practices workflows, and these are the recommendations from the GATK method development team on what is the best way to analyze this data to get the most out of your sequence data. And we're going to, so there's different workflows for the different types of variants we want to identify. And we're going to distinguish kind of 3 phases of analysis, which are kind of common across all of them. Data pre-processing, we're going to go from that raw sequence pile to having actually aligned the reads to the reference and also cleaned up to get rid of or mitigate the presence of technical artifacts. In the next phases, we'll do the variant discovery process proper, or we'll apply some kind of algorithm to identify the variation. And in the third phase, which is kind of optional and very project dependent, which I will not go into detail on, that's where you might do some additional filtering, some additional annotation, and things like that. But let's focus on data pre-processing and variant discovery for now. So what processing needs to happen? First, we need to align each read to the reference genome, okay? Because the read is no use to us if we don't know where it aligns to, if we don't know what region it informs us about. So we're going to use a genome mapper, BWA or STAR, depending on the type of data we're working with. And we're going to identify for each read where it aligns. All right? Now, I mentioned that at Broad we have a slightly special workflow. We use a format that's called unmapped BAM instead of FASTQ. It just means that it's a slight format difference. It involves a slightly more elaborate processing workflow. Hopefully, you guys should not have to care about what goes on in here. The key takeaway is that this approach allows us to associate the metadata that belongs to, that qualifies the sequence within the files with the sequence, as opposed to having it separate. And it's just from a data management point of view, it just decreases the chance of something going wrong. But again, this is something that we have more documentation for on the website in the unfortunate event that you actually need to know about it. All right, the format that is going to be output by the aligner is called SAM or BAM for sequence alignment map. BAM is just a binary compressed alignment map. It's the same information, but written a bit differently in the file to take less space. And this is how one record will be structured. You will have the read name, which we already had from the sequencer. We have the sequence and the quality scores. Both also come from the sequencer. But now with the mapper, we've added the position in the genome. We've added the quality of the alignment, mate information, assuming we're working with mate pair data, and the cigar string. And the cigar string is, it stands for, do I have a, no, I don't have that in this deck. I always forget what it stands for. Somebody really wanted it to spell cigar. It's just a compact representation of the structure of the alignment that says I have 23 matching positions, then I have, this is RNA-seq data. I have 6 ends, meaning I have a splice junction here, and then I have 10 more matching positions. You can also describe presence of indels and so on in that. And the point is just that it's a compact representation. With just this record, you can represent how that read sequence aligns to the reference. Okay? And that's the information you're working with for the rest of the pipeline. Now, when you have that, you have your aligned data, you have to go through a few hoops of correcting for technical artifacts. The first one you're going to correct for is duplicates. Duplicates can arise in several different ways during the sequencing process. They're basically reads that look like different reads, but they come from the same original fragment of DNA, so they're non-independent observations. And because all our algorithms depend on only processing independent observations of data, we need to eliminate those. So when we recognize we have a stack of duplicates, we're going to just keep one representative one and get rid of all the others. All right, so that's to mitigate the presence, the effects of duplication. Next, if we're working with older data sets, especially, and depending on the variant colors we're going to use, we may, in some cases, need to run a process called indel realignment, which is going to realign reads to rationalize the sequence representation. This is not something we need to do anymore with the newer versions of the variant colors, but it's something that used to be used a lot and is still used in some of the somatic workflows. It's called co-cleaning, if you've heard of co-cleaning. That's when you process your tumor and normal BAMs through indel realignment together. Base recalibration, BQSR. This is a processing step that is going to help us deal with the fact that the sequencer makes mistakes. I mentioned earlier that the sequencing process is not perfect, despite what the company reps might claim. The thing is, we find the systematic biases in some cases in how the sequencer assigns qualities. Sequencers can sometimes make mistakes in a systematic way. Random mistakes are fine. We can deal with random. But systematic means that we're going to have accumulations of evidence that are spurious due to artifacts. And so this process is going to apply machine learning to recognize when that happens and mitigate it. And if we're working with RNA-seq, I just want to point out we're using a different mapper, and we're using a different, we're adding a processing step that's going to deal with the presence of splice junctions in RNA-seq. Because in RNA-seq, if you think about what you're sequencing, which is the spliced mRNA, you've lost that sequence there from the intron. And so where a regular genome mapper would think that you have a massive deletion here, your RNA-seq mapper knows that, no, it's just a splicing junction. It doesn't have to freak out. Okay? So that is a processing step that we apply if we're working with RNA-seq. And at that point, you have a BAM file of sequence that has been aligned to the reference and is ready to go, ready to use for variant discovery proper. Okay? So let's look at the variant discovery workflows. I'm going to look at 3 main use cases that are kind of the ones we have in working order right now. We have additional cases that are under development. Germline SNPs and indels is the one that we have the most experience with, but we've been developing a lot towards the somatic short variants and the CNVs. And we're quite excited to have those workflows now in GATK4, which are available in a beta state if you want to play with them. So let's go over the germline SNPs and indels. And I'm going to focus specifically on the particular challenges of these workflows. And I think the germline short variants might be the most relevant to this audience. The challenge here is that you want to analyze cohorts, not single samples in isolation, for various reasons, both technical and scientific. And I want to focus on the technical aspects. From the technical point of view, if you take just individual call sets, that means just a list of variants from a bunch of individuals that were analyzed separately, and you try to analyze that together, you're missing a lot of information that's going to cause your analysis to be underpowered. Whereas if you can take all the sequence data from all those individuals and analyze it jointly, you're going to have much more power to discriminate the quality of your variant calls. One of the reasons is that when you have a difficult site where, for whatever reason, there was not a lot of sequence data produced, it's difficult for us to make a confident call for any single sample. But if we have data from multiple samples, then we can find corroborating evidence across samples. And if we find evidence of the same sample in variant allele in multiple samples, that tells us that we can be a lot more confident in the fact that it's real as opposed to an artifact. It also helps us get information, all the information we need at all the sites. Because let's say you're looking at these two sites that are predictors of Alzheimer's. In this sample, we have great sequencing quality, we have full coverage, and we're reasonably confident that this person is not at risk, they don't have the variants. This person we don't know because we don't have, something went wrong, we don't have enough information. If we call them in isolation, we're not going to have a variant record for either of those. We're not going to have statistics about that site for either of those. But if we analyze them together with other patients that include people who are essentially the positive controls of the experiments, we will get a record emitted by the program that will contain all the statistics for all the people in the cohort. And that will give us the ability to make an informed decision about whether this person needs additional tests or not. Okay? Now, traditionally, the way you would do that is that you would take all the samples, all the BAM files, and put them through variant calling together. And that's great in principle, but it scales very poorly. It's really hard computationally to do that. And also, because you have to run the program on everything at the same time. Let's say you have 50 samples, you analyze them, great, you get results, but then you have a 51st sample. You have to redo all that work again. And 50 samples is fine, but imagine you're Dano MacArthur and you have 100,000, right? It's just not tractable. So what we do is we break down the variant calling process to, instead of trying to cram all the data through the program in one go, we identify the fundamental information that we need to do the joint calling, but we do that per sample so that then, and we record that as an intermediate file. We basically compile a database of genotype likelihoods in the sense that we identify what are all the likely genotypes for all our samples separately, then we analyze that together. And that second part scales much better as a result. And so this is what enables us to go from, to scale from a few hundred samples up to tens of thousands of samples and above, okay? So that's kind of the key thing to understand about the germline SNP and indel workflow, is that there's this breakdown of different steps that is made to scale up. And what the program is doing is looking at the evidence in each of the reads to evaluate whether there's presence of variance or not. Is that clear? Does that make sense? Okay. In the somatic side of things, it's a bit different because what you're dealing with there, the biggest problem is not scale. The biggest problem is the heterogeneity of the tissue that you're working with. Because if you think about what you get from a tissue biopsy, when you're looking at variant discovery, looking at 4 drivers of cancer, you're getting a whole mix of different cell populations, some of which are healthy, some of which are in different stages of somatic mutation, but with different mixes of mutations in the different cell populations. And as a result, it's technically much more difficult to identify, to tell apart the difference between a real variant and something that's just an artifact. Because where in the germline context, we have an expectation of the allele frequency based on the ploidy of the organism. If I'm a diploid, I expect to have either 1 or half and half or 1 for each allele. If I'm looking at somatic samples, looking for somatic variants, that level of heterogeneity means I have to throw out my ploidy expectations out the window. I can't rely on that. And on top of that, there's problems of contamination between, in terms of, if there's any kind of contamination between samples or stuff like that, which in the germline context isn't as big a deal, in the somatic context, it's a huge problem because then the level of noise is on the same order as the level of signal. And so there we have some algorithms that will tease this apart. A key factor in teasing that apart is using a tumor normal pair. So you have a tumor sample and a normal sample from the same person, but hopefully healthy tissue. And you can compare them. And that's a big part of the rationale for analysis. Does that make sense? Okay. Now, looking at copy number is a completely different ballgame in the sense that here we're not looking at the specific sequence of reads, but we're looking at the amount of reads that we have for any particular target site. So here we're going to look at things from the lens of targets, of genomic targets. That typically that's exome targets, or for whole genome, you have to split up the genome in chunks. And you're looking at how much coverage you have, and you're trying to identify a difference in how much coverage you have on average in the genome and localized differences where you may have more, indicating an amplification, or less, indicating a deletion. And so it's all going to be a numbers game of number of reads and normalizing against some control resources. That main control resource is a panel of normals, and you want to have normal samples that were sequenced in the same way, generated in the same way, that you can compare against to try to identify those ups and downs. Now, when you have exomes, because of that problem where you have biases that causes great differences in the amount of coverage that you have between regions, that's where the normalization process is especially important. So at that point, you have your variants. You have called your variants. They're either in VCF format if you have SNPs and indels, whether they're germline or somatic, or you have text files, TSV files of your copy number variation, which identifies just regions, genomic regions, and how much they were amplified or deleted. At that point, you can apply various filtering and annotation approaches. That's very project dependent, and again, that is something you have to figure out based on your use case. Check the documentation for that. Documentation. Finally, a quick word, and then we're done, I think. 5 minutes, okay. A quick word about the analysis platform. So in the data sciences platform, we've built infrastructure to be able to enable people to do this type of analysis without ripping your hair out. Now, the broad operations team runs the pipeline, the production pipelines for you when you get genome sequenced. They go through an automated pipeline that is run by a team in our department. However, you may need to do additional analysis, or you may want to reprocess some things. You may want to run other pipelines that are not directly related to this. We have built a platform that enables you to do that in a self-service way. If you were familiar with Firehose before, the idea is it's Firehose in the cloud, and therefore it's called FireCloud. A quick note, before I show you a slide on FireCloud itself, all the scripts are written in this thing called Workflow Definition Language, WDL. It's a very user-friendly scripting language that you can use to write your own pipelines. And if you want to see what is contained in the GTK best practices pipeline, they're all written in WDL. They're designed to be understandable and readable by actual humans without a CS degree. Now, we work we have a hybrid mode of working on the local Broad cluster, but also on the Google Cloud. I'm going to fast forward to FireCloud itself just to introduce to you the capabilities of the platform if you haven't heard about it yet. The idea here is that there's this layer of infrastructure that you don't really have to care about too much, but basically they're giving you data management capabilities to manage your data in a way that makes it really easy to share it with your team, share it with collaborators, and so on. Methods repository is a collection of workflows and pipelines that you can use or that you can contribute your own to that allows you to share methods in a way that's very portable and that allows you to reproduce other people's analysis without spending days or weeks trying to set up whatever bizarre mix of R packages and Python whatnot they had on their machine. So that really favors reproducibility across teams. And finally, execution. Cromwell is an execution engine that reads WDL and runs that on the Google Cloud for you. So the goal is really to take the guesswork out of running analyses pipelines by providing you with a tractable, user-friendly interface to that. And it's all set up in a way that you can share things and work with people you need to work with on the cloud without having to worry about a lot of permissions and so on. And it's secure too. So FireCloud itself, so this layer is what we call a workbench. Yeah. FireCloud is a portal that gives you access to all these capabilities with a graphical user interface. There's also an API if you want to interact with it programmatically. It does all this stuff, which is complicated, for you without you having to worry about it. And that's just what I want to leave you with. We are organizing workshops to introduce people to FireCloud. I'm sorry, it's a little bit rushed. It's a little addition to what I usually talk about. But I really want to encourage you to check it out because it is designed to help you get work done without having to deal with all the hassle that you would have to otherwise. Thank you very much for your attention. And I hope this helps. Please talk to me afterwards if you have any additional questions.